Hello everyone, my name is Akriti Srivastav, faculty from Department of Biotechnology and Bioinformatics, Government Holkar Science College, Indore, and I am going to speaking to you today's topic biological database. I am going to share with some of the most important idea that have ever been discovered in this era. According to the recent studies, there will be more change, more competition, and more opportunities in the biological industries in the next few years than ever before. And 72% of the people in this room will doing something different within 2 years. So best of luck now. A few minutes ago, in your previous lecture, the speaker gave a thorough knowledge about introduction to bioinformatics. But as a bioinformatician, when I am going to start major topic of bioinformatics, that is biological database, little bit discuss about the concept of bioinformatics in very short way. Okay, so what is bioinformatics? Bioinformatics is an interdisciplinary field that develops methods, software tools for understanding biological data. In particular, when the data sets are large and complex. As an interdisciplinary field of science, bioinformatics combines biology, computer science, information engineering, maths, and statistics to analyze and interpret the data or interpret the biological data. Bioinformatics has been used in silico analysis of biological query using mathematical and different statistical techniques. Bioinformatics include biological studies that use computer programming as part of their methodology, as well as a specific analysis pipeline that are repeatedly used, particularly in the field of genomics. Now, we come on today's topic, that is biological database. So, firstly, I am going to give you a basic definition of biological database. Firstly, biological database is a collection of data which is structured, searchable, updated periodically, and also cross-referenced. In other words, we can say when biological experiments and computational science are merged, they form the biological databases. Actually, the biological database are library of life science information collected from scientific experiments, published literature, high throughput experiments, technologies, and computational analysis. They contain information from research area including genomics, proteomics, metabolomics, and microarray gene expression and phylogenetics also. Information contained biological database includes gene function, structure, localization, both cellular and chromosomal, clinical effects of mutation, as well as similarities of biological sequence and structure. Biological database can be broadly classified into sequence, structure, functional database. Nucleic acid and protein sequence are stored in sequence databases and structured databases store solved structure of RNA and protein. Functional database provide information of physiological role of gene products. For example, enzymes, activities, mutations, phenotypes, and biological pathways, and so on. So now, the breakdown of this lecture is biological data, biological information, and increasing amount of biological knowledge is sorted in databases. And the database is systematically organized collection of data and information. The database can be single file or the collection of files which stand alone or are organized by specialized computer programs called database management system. A database can be single file or collection of files which stand alone or are organized by a specialized computer program called database management system. There are many biological database modes available to the public, generally through the internet. So now, what is the type of storage or data storage sources? As you can see here in this diagram, there are many type of data storage sources. It can be folder, cloud, files, or server. These are different type of storage sources are collectively perform and organize the biological data and store it in a proper manner. Then, what is the structure of database? Every database have a specific structure. It may be file or flat file system, relational database type of data, object oriented, exchange and publication technology based databases. What is the methodology of biological database and what is the process of biological database? The first step is sample collection from the food or the environment. The next one is data DNA extraction, amplification with the help of PCR and electrophoresis, and then gene sequencing process with the help of some new technologies like NGS and Illumina, and then organize a data in different type of specialized bioinformatics databases. Okay, what is the plan of work or the plan of research? First one is plan the research area, which kind of interest of user or researcher, then collect the data according to research, then analyze it, then retrieve from the different sources or from the prior knowledge, then protection or security management, process the data, and lastly transport it in online transport the data from online manners such as publication in general and so on. Here again I am going to show a diagrammatic representation of plan of work. In this diagram or in this slide you can see species sample collection, then data or DNA extraction, DNA sequencing method, then coupling sequence data and scientific name of the sample. In other hand, we can do the species sample with morphological species identification method as well. Then DNA barcode preparation and registration of that particular barcode in related database. And finally, after the submission of finally, after the submission of data or information, we can easily apply the applications of bioinformatics. So, why do we need bioinformatics, or why do we need biological database actually? Large data sets has grown very fast and need to organize and store it properly. Make biological data available to scientists for less time consuming research, and also to make biological data available in computer readable form. By the help of computer readable form, it is very easy to access or retrieve a data and analyze it. Why do we need bioinformatics actually? Bioinformatics is necessitated by the rapidly expanding qualities and complexity of biomolecular data. Bioinformatics provides a method for the efficient storage, annotation, research and retrieval, data integration, and data mining and analysis methods. So finally we come on conclusion, that is bioinformatics is essential for the achieving, organizing and analyzing the data from sequencing, structural genomics, microarray proteomics and new high throughput assays. So in this slide I am going to show the growth curve of different biological data sets such as gene sequenced and 3D structured data sets. Why we study the biological databases and what the need. The first one is collection, or the first one is collected data quickly. Inexpensive methodologies are easily available, easy to do research with the help of computer readable form of data. Few rules are followed for analysis and interpretation. Readily available samples and low cost methods are easily available, which can help to analyze data in very less cost effective manner. So, what is the type of biological data? There are so many types of biological data such as scientific literature, genome sequence, nucleotide sequence, genes, genetic variation, protein sequence, molecular structure, biochemical pathway and chemicals, disease and taxonomies, and some other phenotypes data. You can see here in another diagram of major type of bioinformatics data. It shows there are again various type of biological information or data available today. It can be literature or ontology, gene expression, protein sequence, protein structure, chemical entities, genomes, DNA and RNA sequence and structures, protein family like motif and domain information, protein interactions, pathways and systems. These all are the major type of biological or bioinformatics data. So, what is the goal of bioinformatics data or the biological data? The major goal of bioinformatics data are integrated sequence 3D structures, expression patterns, interaction and function of biomolecule to gain a deeper understanding of biological mechanism, process and system. The second one and the important goal is bioinformatics aim to bridge a gap between data and knowledge. With the help of bioinformatics, we reduce the gap between data and knowledge. Now I'm going to explain some of the research areas of bioinformatics before we discuss about the major biological databases. Bioinformatics research area includes but are not limited actually organization, classification, dissemination and analysis of biological and biomedical data, biological sequence analysis and phylogenetics. Next one is genome organization and evolution. These are the major topics or the major research area. What is the type of biological database? According to the theory of central dogma, we are majorly divided the biological information or biological database in two different types. First one is nucleotide database and next one is amino acid databases. According to the theory of central dogma, DNA are transcribed into RNA and RNA are translated into protein, and with the help of this process we easily collect the data in the form of nucleic acid or amino acids. Now, where did the data comes from? Bioinformatics arose a molecular biology began to be transformed by the emergence of molecular sequence and structural data. So now we can discuss the recap about the information. The key dogmas or the central dogmas of molecular biology. The first point is DNA sequence determine the protein sequence. Next one is protein determines a protein structure information. Then protein structure determines protein function and it related with the motifs region or the conserved information. Then the third one is protein structure determines protein function. And last one is regulatory mechanism determine the amount of particular function in space and time. So we come on the conclusion is bioinformatics is now essential to achieving, organizing and analyzing the data related to these processes. So what is the database? Computerized store of data that is organized to provide efficient retrieval. That means user standardize the data records or formats to enable computer handling. Or discussed previously, there are different type of structure of database such as flat file or relational database and object oriented database. These all are the different type of standardized record format system to organize the data in particular manner. So key databases features allows for first is adding, changing, removing and merging the records. The next one is user defined query and extraction of specified records. So what is the desirable feature includes? First, contain the data you are interested in. Then allow fast data access and provides annotation and curation entries, and then provide link to additional information, possibly related or linked databases, then allow you to make discoveries or new knowledge discoveries. Now we discuss about the quality of databases. The first one is redundancy, checking data, human curation, and updating data. These all are the essential quality of the biological databases. So what is redundancy? Data redundancy occurs when the same piece of data or information is sorted in two or more separate places and is a common occurrence. How does data redundancy occurs? Sometimes data redundancy happen by accident, and while other time it is by intentionally. Accident data redundancy can be result of a complex process and or insufficient or inefficient coding, while the intentional data redundancy can be used to protect data or ensure the consistency simply by leveraging the multiple occurrence of data from disaster recovery and quality checks. If the data redundancy is intentionally, it's important to have a central field or a space for the data. This allows you to easily update all records of redundant data when necessary. When the data redundancy is not purposeful, it can lead to variety of issues which we will discuss some other day. The next one is checking data. Of course, it's very important to check data or validate data before we submit or store the information in database. Then next one is human curation. What is data curation or human curation? The data curation is organization and integration of data collected from various sources. It involves annotation, publication, presentation of the data such that the value of data is maintained over the time and the data remains available for reuse and preservation also. The data curation includes all the purpose needed for principled and controlled data creation, maintenance and management, together with the capacity of the added value of data. In science, data curation may indicate the process of extraction of important information from the scientific text, such as a research article by experts, to be converted into the electronic format such as biological database. Okay. The last one is updating the data. It's very necessary to update a data time to time. Our next point is maintained by. Who maintain the biological database actually? Large public institutions like EMBL, EBI and NCBI, and also some academic groups and scientists and some other commercial companies. These all three different type of data maintaining organization which maintain a data. So here in this slide you can see different type of biological database. In this slide you can see different type of database. The number of database nowadays the number of database is very huge, because time to time the specialized database are formed rapidly. There are lots of bioinformatics database. For a annotation listing of major bioinformatics database which we can study or we can see today. Some major molecular database are: the most popular bioinformatics database focus on biomolecular sequence such as GenBank, UniProt, DDBJ, PIR, and so on. Biomolecular structure like PDB database, vertebrate genome database like Ensembl, small molecules like PubChem, biomedical literature, PubMed. These all are some popular bioinformatics database. There are also many popular database which are used as a specialized database also. First one is classifying protein family or domain and motif, such as PFAM, ProCite, Print. These all are also a secondary database type, which depends the study related with the alpha and beta sheets of the protein. Next one is a specific organism like fly based or worm based. These databases are highly specialized for particular species or organism. Next one is a specific protein of biomedical importance like kinase DB. It's a kinase database. And GPCR DB. It belongs to GPCR or signaling database. Next one is a specific disease or mutation related database like OMIM. OMIM are related with online Mendelian inheritance database. The biological database, in this slide you can see biological database are classified into 3 different types: protein database, nucleotide database, and composite database. Protein database are again classified into primary, secondary, and tertiary databases. And nucleotide database belongs to GenBank, EMBL, and DDBJ. Some composite database like OMIM, NCBI, and PubMed are also available. When we discuss about the source, example of the sources, the primary sources are research article, peer review journal, clinical trials, interviews, correspondence, patterns, lab notes, data sets, thesis and dissertation as well. And the secondary sources are summaries, interpretation of primary data literature, review articles, textbooks, dictionaries, and encyclopedia. These all are also example for tertiary sources of information as well. Now we discuss about the different features of databases, such as firstly we discuss about primary database. Primary database also referred to a archaeal database. It includes raw nucleotide like DNA and RNA and protein sequence data, protein structure data, microarray data, may be annotated or curated informations, original biological data from experiments, and primary sequence data from DNA, RNA, and protein sequence. Okay, firstly we discuss about the primary database. According to type of data, there are basically 2 types. Firstly, we discuss about the primary database. According to type of data, There are basically two types of primary database: primary protein database or primary nucleotide database. GenBank DDBJ are the nucleotide database, and SwissProt and PIR is a protein database. The primary database, also known as repository or archival database, are highly organized and user-friendly gateways. A huge amount of biological data produced by researchers around the world and organize it into the primary database. Primary database are the product of conceptual translation of gene and genome determined using DNA sequencing. Primary database also, they are populated with experimentally derived data such as nucleotide sequence and protein sequence. Experimental results are submitted directly into the database by researcher and scientist groups, and the data are essentially archival in nature. Once database gives a accession number, the data in primary database are never changed. They form a part of scientific records. Some of the example of primary databases is PIR, Protein Information Resource, SwissProt, GenBank and EMBL. These all are the primary database. Now we discuss about some of the major biological database. You can see here, first one is NCBI. The NCBI or National Center for Biotechnology and Information at National Institute of Health was created in 1988 to develop information systems for molecular biology, in addition to maintaining the GenBank nucleic acid sequence database, which receives data through international collaboration with DNA Database of Japan and European Molecular Biology Laboratory, that is EMBL, Nucleotide Sequence Database, as well as from some scientific community. NCBI provides a data retrieval system and computational sources for the analysis of GenBank data and many other kind of biological data. For the purpose of, you can see here in this slide, NCBI includes some of popular resources such as like PubChem, Bookshelf, PubChem Central and PubChem Health. Some of biological analysis tool like BLAST. This is a basic local alignment search tool used for alignment methods, and some biological databases like nucleotide database, genome, SNPs, gene, protein and PubChems. So with the help of NCBI, we can easily access different type of data sets in same platform. That's why we can say the NCBI is a collaborative database. You can see here the list of, for same query, here the list of literature database as well as gene, protein and genome database, and also clinical and PubChem, that means chemical databases. This slide shows the retrieval information from the NCBI. The blue line shows the definition, and then you can easily see some numerical information like 548 amino acids. It's a range of amino acid or the length of particular sequence, and then some Hyperlinked databases like PubChem and taxonomy and graphics, these all are interrelated or correlated with same platform. NCBI, there are some NCBI metadatabases like Gene for molecular data and literature related to genes. Homolog genes, this database are related with annotate collection of homologous genes from selected eukaryotes. Similarly, taxonomy database related with the organism taxonomic classification. PubChem is related with the small organic molecule and their biological activities. And last one is biosystems. It relates with biological chemical pathway and process linked to NCBI genes and gene products, a small molecule and structures as well. So the next one is GenBank. GenBank is NCBI primary nucleotide only sequence database. It's archival in nature, reflect the state of knowledge at time of submission. It's again subjective, reflects the submitter point of view. Redundant, it can have many copy of the same nucleotide sequence. GenBank is actually 3 collaborating international databases from the US, Japan and Europe, that is GenBank, DDBJ and ENA or EMBL. You can see here, these all 3 are synchronized with each other and share the data weekly. The underlying raw DNA sequence are identical. The different sites provide different view and ways to navigate through the data. And then access to GenBank is typically through Intrez. With the help of Intrez, we easily access the nucleotide data and in a readable form. Next one is PIR. PIR, Protein Information Resource. It's an integrated database. The PIR is collaborative endeavor between PIR, MIPS and JIPID. PIR is now a comprehensive, non-redundant, expertly annotated and object relational database. The unique feature of PIR is it maintains the classification of protein sequence database on the superfamily concept, and also homology domain may correspond the evolutionary building blocks, while sequence motif represent functional site or conserved region. The PIR maintain the classification approach allows a more complete understanding of sequence classification or sequence function structure relationship. This is the output screen of PIR database. By the help of PIR, we easily studied about different type of classes or family of protein. Now we come on secondary database. Secondary database comprise data derived from the result of analyzing primary data. Secondary database often draw upon information from numerous sources including other databases like primary and secondary, control vocabulary and scientific literature. They are highly curated, often using a complex combination of computational algorithms and manual analysis and interpretation to derive new knowledge from the public record of science. For example, ProFile, ProCite, PFAM, and InterProScan are the secondary databases. Secondary database, also referred a derived database. See, these are the example of secondary database: ProCite, PFAM, Print, and Pattern. Next one is protein structure database or tertiary database. In biology, a protein structure database is a database that is modeled around the various experimental determined protein structure. The aim of most protein database is to organize and annotate the protein structure, providing the biological community access to the experimental data in a useful way. The data includes in protein structure database often includes three-dimensional coordination as well as experimental information such as unit cell dimension and angles for X-ray crystallography determined structure. Here you can see the home page of PDB. The protein database is a database for three-dimensional structure data of large biological molecules such as protein and nucleic acid. The data typically obtained X-ray crystallography and NMR spectroscopy and increasingly cryo-electron microscopy techniques, and submitted by the biologist and biochemist from around the world, are freely accessible of the internet via the website of the members organization like PDBE and PDBJ and also RCSB. Here you can see in your slide, this link comes from RCSB organization. The PDB is a key in area of structural biology such as structural genomics. Most major scientific journals and some findings as agencies now requires the scientists to submit their structure data to the PDB. Many other databases use protein structure deposited in the PDB. For example, SCOPE and CATH classification protein database. Now we come on another database, that is PubMed. PubMed is a freely search engine assessing preliminary the Medline database of references and abstracts on the life science and biomedical topics. The United States National Library of Medicine and the National Institute of Health maintain the database as part of the INTRAS system of information retrieval. From 1971 to 1997, online access of Medline database had been primarily through institutional facilities such as University Library. PubMed first released on 1996. Okay, here you can see the home page of PubMed. In this home page, it gives us so many options like overview about the PubMed database, then PubMed Central, it's another type of literature database, advanced helps, and news in PMC. PubMed is a very essential database just because of PubMed delivers a publicly available search interface for Medline as well as other MLM resources, making it a primary resource for biological literature and one of the most widely accessible resource in worldwide. The general difference between PubMed and Medline is that Medline is the curated collection of citation of article published in journal which have been approved by NLM and have been indexed by the NLM to have MESH term associated with those articles. Now we come on specialized database Go ontology. There are three ontologies in Go: biological process, molecular function, and cellular component. In biological process, a commonly recognized series of events like as cell division and mitosis. In molecular function, an element, activity, task or job like kinase activity and insulin binding activity. In cellular component, where a gene product is located. For example, in mitochondrion or mitochondrial membrane. Actually, the gene ontology or GO is a major bioinformatics initiative to unify the representation of gene and gene products attributes across all the species. More specifically, the project aims to maintain and develop its control vocabulary of gene and gene products. Then the second one is annotate the gene and gene products, provide a tool to easy access to all aspects of data provided by this project, and to enable the functional interpretation of experimental data using the Go or gene ontology. Now we discuss about some issues of database. First is interoperability. The file format, because the file formats are different, such as different header details, different field names and representation of sequence type, it increases the interoperability. Some access issue like infrastructure opening, system opening या computing system, distributed networks, and user interface and web interface, and some command line issues. These all are the basic issues with databases. And also I'm going to discuss some errors in sequencing and gene structures like sequencing read errors, contaminations, mistyping, and incorrect assignment of coding sequence, or incorrect assignments of exon and intron boundaries. Similarly, some errors or inconsistency in annotation like interference and annotations made on the basis of incorrect similarity comparison. And the next one is propagation of error from incorrect references, incorrect or different spellings. So these are the issues and errors in databases. After the discussion, I am going to summarize all the discussion. So what is the summary of the lecture is: bioinformatics is a computerized or computer aided biology. Bioinformatics deal with the collection, organizing and interpretation of wide range of biological data. There are large amount of primary, secondary and tertiary biological databases available now. The next one is the NCBI, SwissProt, EBI are the major online bioinformatics database, and we also explore these database little bit. Last one is also introduce GenBank, PDB and PubMed database as well. Thank you for all. Okay, thank you all for listening me. If you have any query, please send me your query to given mail ID, and also we are sending you a link for quiz. Please complete the quiz and submit it before 4:30.